All right, we're going to go over the answers to the questions for case studies number 25 and 30. Let's go ahead and start with case 25. It started out with question 1, just having you define some terms. Of course, neutropenia is a lack of neutrophils. GCSF, which can be a little bit of a mouthful, is a granulocyte colony stimulating factor. It's a cytokine, and that cytokine is a ligand that binds to GCSFR, which is the GCSF receptor. When GCSF binds GCSFR, then signal transduction results in gene expression, which triggers the differentiation of granulocytes. And then we also have heterozygous mutation, which would be a mutation of only one allele. And then also allogenic stem cell transplantation, which is a transplantation of stem cells from a donor. It could be a family member, an acquaintance, a complete stranger. All right, now question 2 asks about primary and secondary granules of neutrophils. How do they differ? How are they similar? So primary granules are antibacterial compounds, things such as myeloperoxidase, acid hydrolases, defensins. And secondary granules also have antimicrobial properties. They contain enzymes, but in addition to that, they function as complement activators. So how long does the average human neutrophil survive? It's around 5 days. And how does the body replace neutrophils? Neutrophils are derived from myeloid progenitor cells, which come from hematopoietic stem cells. And a normal neutrophil count for an infant would be around 1,500 to 4,500 neutrophils per microliter. Question 5 asks about why the doctors thought that the neutrophil issue they were seeing in their patient was not due to the umbilical cord infection, and that's because after the infection resolved, the counts didn't recover. They followed that up with trying to figure out what it was that was going on with their neutrophils, which can also be considered part of question 6, which asks how do you differentiate between peripheral destruction of neutrophils versus decreased production. And what they did was they did a bone marrow aspirate, and they saw that there was a lack of mature neutrophils in the bone marrow. So that led them to know that what they were seeing was decreased production of neutrophils due to neutropenia. Another thing that could have been done if they had not seen that in the bone marrow aspirate would be to label the neutrophils and check for their survival. Now, why do you think severe combined neutropenia is not treated with neutrophil transfusions? Neutrophils are very short-lived, of course. We already said they last around 5 days. That would be too many transfusions. Also, if you think about, it would have to be so many cells because there would have to be a level maintained just in case an infection did take place. And with this high turnover of neutrophils, it actually can cause an inflammatory response or cytokines that could be released that can actually trigger inflammation. Question 8 asks about some ways that somatic mutations in the GCSF receptor gene might cause leukemia, leukemia being an excessive number of a type of cell. So mutations could lead to a receptor that isn't dependent on ligand binding. So this is a receptor that would be always on. which would then cause increased or rampant proliferation. And also, the GCSF receptor is removed from the cell membrane and digested via ubiquitin tagging. And if there's a mutation in the ubiquitination site, then that means that the receptor is not going to be digested. It's going to stay in the cell membrane. Why might patients have low neutrophil counts but have high monocyte and eosinophil counts? Want to consider the fact that all these cells are derived from the same progenitor cells, and if you knock one down, there could be compensation, which could result in an increase in these other cell types. All right, now we're going on to case study number 30, which also started with having you define some terms. So we have, the first term is consanguineous, like a consanguineous marriage, which would be the marriage of people who are closely related to each other. Haploinsufficiency is where there's only one functional copy of a gene, and the presence of just that one copy of a gene results in insufficient expression, and so then the phenotype isn't normal. So the fact that there's only one copy of the gene, depending on whatever it is that gene is responsible for, the expression results in an atypical phenotype. The next one is fully penetrant, and that's the idea that if you have an allele, you get a phenotype. Or if we're talking about a disease, if you have the allele for a given disease, you get that disease. There's no other factors that are going. It's not like an age-dependent disease. It's not something. There isn't any sort of an epigenetic factor or marker that needs to also come into play for that phenotype to happen. So fully penetrant, if you have the allele, you get the phenotype. And a conjugated vaccine. So that is going to be a vaccine where you have a weak antigen, like a polysaccharide, that doesn't elicit a strong immune response, and it's covalently linked to a strong antigen, such as a carrier protein. We'll talk about that a little bit more further in or further down with one of the other questions. So what is a normal white blood cell count for a 3-year-old? That would be around 4,000 to 12,000 cells per microliter. And question 3 is asking about why it is that patients with congenital asplenia, why do they get diagnosed after pneumococcal infections versus other infections? And what that has to do with is we're talking about these are encapsulated bacteria that cause these types of infections. And these encapsulated bacteria are not easily cleared by the innate immune system. And we actually need marginal zone B cells that are located in the spleen to mount a response against this sort of an infection in order to clear it. So other infections are cleared without incident, you could say, by the immune system, by the innate immune system. But these encapsulated bacteria actually require these specialized B cells to respond to them. All right, question 4. So why do patients with asplenia need to get vaccinated against these listed bacteria, or what's special about these specific types of bacteria? So these are also polysaccharide-coated or encapsulated bacteria. And as we talked previously, there's the need for vaccination against these types of bacteria because no spleen means no marginal zone B cells, which means that a vaccine is needed. All right, question 5. So when patients are immunized against those same bacteria, they're usually given a protein conjugated vaccine subcutaneously. And the reason for the conjugated vaccine is that, or a little bit more specifically, we talked about this a little bit previously, but the B cell that is going to respond to the vaccine is going to recognize the polysaccharide portion of that vaccine. So the antigen that's actually part of that polysaccharide coat, or that polysaccharide capsule that's actually on the bacteria. And then a T helper cell that's going to be involved in the activation of that B cell is going to recognize the protein epitope that comes from that protein carrier that's going to be presented to the T helper cell. So it's the co-stimulation of that B cell by the polysaccharide portion of the vaccine, and the T helper cell recognizing its protein epitope that's going to result in an effective immune response. And the reason why it's given subcutaneously is that that way a dendritic cell will actually encounter the antigen and take the antigen to the lymph nodes where that stimulation can take place involving the B and T cells. All right, number 6. Let's go ahead and look at another version of this question. And it is, your nephew ruptured his spleen. He has a spleen removed and suffers no other long-term damage. His parents, one of which is your sibling, don't want to have him vaccinated. They claim that the spleen just filters the blood. What would you advise them besides vaccines? What else would you tell the parents to do for your nephew, their child? So you will want to inform them that the spleen is not only a filter of the blood, but it's also involved in fighting infections of the blood. Of course, depending on their understanding and curiosity, you can explain that a little bit more, but the basic idea is that no, that's not all the spleen does. The spleen actually has, a role in the lymphatic system and a role in fighting infections. And so because of that, and because of like what was seen earlier, certain bacteria, these polysaccharide coated bacteria, we actually need a spleen because we need those marginal zone B cells. Since your nephew no longer has a spleen, your nephew no longer has those B cells, so he needs to be vaccinated against those polysaccharide coated bacteria types. And then another thing that would be good to recommend to them would be, the use of prophylactic antibiotics, or, preventative antibiotics before teeth cleaning operations. Any thing that's going to have a good chance of resulting in the exposure of the blood to bacteria. Okay, and question 7. Okay, so the key to this question is that the reason why the Vanderveer family is pretty unique, even more unique than having this asplenia in their family, is that it wasn't genetically dominant. So the inheritance pattern of the disease in this family was autosomal recessive. And then, of course, with an autosomal recessive inheritance pattern with a larger sample size, of course, you would expect around 25% of individuals to show or express that phenotype, or 2 out of 8. And that's it. I'll see you on the next one.